Hi friends, so these are the IB Bio class notes for class today. You should be watching these in class on Friday, March 16th or Monday, March 19th, and I hope that this allows us to continue to move forward with the content despite the fact that I'm not there with you, and I hope that this will kind of fill in some holes or give you a second exposure, give you another opportunity to interact with the content as we continue to march on through this content in the next month or so, because we are getting closer and closer to test time. So we need to march through this content to make sure that everyone is as prepared as possible for the IB exam and prepared to do well. Note that this video will be removed from the YouTube feed, but the PowerPoints will go up on Blackboard, and there are just some issues tied to that. So I'll do what I can to make sure you have access to this, but I can't guarantee that you're going to have this video. So I would highly recommend that you spend some time now to watch this, take notes, re-watch it if you need to. But as another note, there's on the class log for today up on Blackboard, there is an answer key to the genetics problems that you received in the previous class. I would double check your answers if you tried that problem set with the answer key to see how you did, and if you didn't get the same answers, check with a classmate, work with somebody before you watch this video, because this video is going to add some more complication to those practice problems, okay? And then there's another set of practice problems for after this video set that you'll get from the substitute, okay? So let's go ahead and get started here. So this first target for these notes is the 4.3.5, says explain how sex chromosomes control gender by referring to the inheritance of X and Y chromosomes in humans. Now remember, from the karyotypes, it's actually the last set of homologous pairs, X and Y, or X and X, that are the sex chromosomes, and these are the two chromosomes that control gender. So all males, unless there's some kind of chromosomal disorder that some of you researched, are going to be XY. All females are XX. Again, unless there's some chromosomal issue tied to non-disjunction. So those are the chromosomal genotypes, I guess, of this gender. But notice that when we talk about these chromosomes, we're actually talking about thousands of genes that are tied on those chromosomes, and we'll come back to that in a couple minutes. But when we refer to gender, when we talk about a Punnett square, we actually are talking about chromosomes rather than the individual genes like you did on the practice problem set. Now, I just want to review very quickly the definition of homologous chromosomes. So these are chromosomes that code for the same gene, but can be, they don't have to be, but can be different alleles. And one of these, for each person, one came from his or her mom, and the other one came from his or her dad. So one of these X chromosomes in this woman came from her mom's egg, and the other X chromosome came from her dad's sperm. For this male, this X chromosome came from his mom's egg, and the Y came from his dad's sperm. And when we talk about these homologous pairs, X and X are homologous pairs. So if we imagine a little dotted line down the middle as if these were sister chromatids, we don't typically draw those when we talk about this because those sister chromatids are exact copies. So to simplify it, we show where there's the potential differences. So X and X are the same, but X and Y are different. And this is really the only case where the homologous pairs don't match up in size when we're talking about the X and Y chromosome. But we typically draw Punnett squares for this kind of relationship, even though we're not talking about individual genes on the chromosomes. Remember, there are thousands of them along the chromosome. We actually talk about the entire chromosome moving. which is really actually a good thing to talk about, because it's the chromosomes that move into the sperm and the eggs during meiosis, and with those chromosomes they carry thousands of genes. So some genes travel together, especially if they're right next to each other on these chromosomes. The one thing that might mess that up would be crossing over. And if crossing over occurs, then some genes that might be associated typically on the same chromosome, let's say here and here, may get split up if this part gets cut off and the other chromosome had a different allele for the same gene. But we're talking about the entire chromosome moving, so we can talk about the X chromosomes and the Y chromosomes. We've got the mom's X chromosomes here. These are the homologous pairs. They've split apart. So let's imagine that this is in one egg. This is another potential egg. This is coming from dad, so this is a sperm carrying this X chromosome. This is a Y carrying the X chromosome. Now remember that these sperm and egg are carrying more than just this X chromosome. They're carrying 22 other chromosomes in humans. But when the sperm and the egg come together, as far as the sex chromosomes go, which control gender, X and X come together in this situation, X and X come together in this situation, XY and XY. So really there's a 50% chance of every time a sperm and egg come together, every time fertilization happens in humans, 50% chance of it being a boy and 50% chance of it being a girl. I hope you see that, or whether that's a review from 9th grade. But here, if one of these X's and the X from dad come together, you get a girl. And if the Y from dad and one of the X's from mom come together, you have a male, a boy. And that's what's happening. Now let's focus in on the differences here between the X and the Y. So some genes are present on the X chromosomes and absent from the shorter Y chromosomes. So obviously the X chromosome is much longer than the Y chromosome, which means that there are more genes on it than the Y chromosome. And we're looking at the Punnett square that we just drew. We know that the gametes, or the sex cells, or the sperm from the father, determine the sex of the offspring, because it's only dad that has the Y chromosome that could potentially be passed on to the offspring. And if the offspring gets a Y, it's going to be a boy. So dad really has a control here, which is kind of interesting when you think about the history of King Henry the VIII, I believe, and all the wives that he had, and that he blamed them for not giving him a son, when really, in fact, it was his sperm that were not making it, actually the sperm with the Y chromosomes that were making it to the eggs. And why that happens, I don't know, but that's the case. And let me go back. Let me just point out, and remind you that this is for every interaction between sperm and egg. So that doesn't mean that if you have a boy, that the next child you have is a girl. And I hope you realize that, and that's why we see families that don't have equal distribution of gender in the offspring. So you could have 2 sisters, 2 brothers, more than 2. You could have 3 sisters, 5 sisters. It's because every time a sperm and egg come together, it's like flipping a coin. Some of these sperm, one of the sperm is going to make it, and half of them have an X chromosome, and half of them have a Y chromosome. So it's equal odds every time fertilization occurs. Okay. So let's move on to sex linkage. So essentially this is associated with gender, because we're talking about genes that are on the sex chromosome. Now, this is a definition that you need to memorize. I'm afraid that many of you are not committing these terms that IB wants you to define to memory, which is leading you to get questions wrong on assessments. For example, on the last quiz, many of you wrote down that a gene is an inherited trait. Well, genes are not traits. Genes are sections of DNA that code for traits. So there's a clear distinction there, and IB is not going to give you credit for that. So you need to make sure you commit definitions to memory. These are easy points to get, easy marks, to help boost up your scores. It's also impacting your grading class, which is quite significant for some of you at this time. But let's look at these a little more closely. Most of these are X-linked, which means that we're talking about genes that are on the X chromosome. And let's talk about recessive. So X-linked recessive. That means that if someone has the recessive allele on their X chromosome, they can be affected. So men can be affected and women can be carriers or affected. So for women it's a little more complicated. So let's actually, before we talk about this diagram, let me break this down a little bit. So we're talking about X-linked recessive, which means that for males, remember, that are XY, to be affected, they need the recessive allele, which means that they have to be, let's use the lowercase h, so they have to be X little hy. And this little h is referring to a gene on the X chromosome. So this male has the recessive allele, which means he does not have the dominant allele, which means that the recessive allele will show, is affected. The unaffected male will look like this. And if you think back to the Punnett squares we were just drawing, this X little h, or the X big H, were contributed to this boy or man from his mother. For women it's a little more complicated. Remember, women are XX. So if we look a little more closely, an affected female will be X little h, X little h. Remember that one of these X little h's came from mom, the other one came from dad, which means that if dad contributed this X little h and dad only has X and Y, then dad must have had this condition as well. Females can also be carriers, which mean that they carry the dominant, they carry the recessive, but it's hidden. So to have the condition, they need to have the recessive trait expressed. They have the recessive allele in this case, X big H, X little h, but because there's an X big H, which is the dominant allele, this dominant allele will mask this one, and it potentially could be carried to the next generation if the next generation received an egg, or started from an egg that received this homologous set, this homologous chromosome of this X that carries the recessive allele H. Or women can be unaffected, which I think you can figure out is homozygous dominant. Okay? So let's go back to this diagram. If I can navigate back to it. Okay. So men can be affected, men and women can be carriers or affected, and both of them can be unaffected. So let's look at this diagram. This male is unaffected. So therefore he's got X big H Y. Mom is a carrier, so that means we'll do this big H Y. And mom is a carrier, so she's X big H, X little h. Now when you do a Punnett square of this, let's put dad on this side this time, and mom up here. Mom's a carrier. And we combine these, and remember that these on the left would represent sperm, these on the top would represent the eggs, and these are the results of meiosis. Inside each Punnett square would be a potential fertilization, or the new diploid that was formed from fertilization. So if we look at this, if the chromosomes combine to form, X big H, we have an unaffected female. If it's X big H, X little h, we have a carrier in the next generation. We have X big HY, it's an unaffected male. Or if we have X little hy, we have an affected male. So we have, there's a 50% chance that the offspring will have the recessive allele, and if it's a boy, he will be affected, and if it's a girl, she'll be a carrier. And that's really what we see here in these odds down here in the bottom. Okay? Alright, so let's continue. So color blindness and hemophilia are examples, or two examples of sex linkage. And we just use, when I use those letter H's, we could be talking, and that's what I was doing, was talking about hemophilia. And in case you don't know what that is, this is a condition in which the body doesn't make a particular clotting factor, which means that if you get cut, you won't stop bleeding. Now, hemophiliacs can make it to the hospital and get clotting factors injected, but as you can imagine, it's quite problematic. Or color blindness, and because males can either be affected or not, and females are carriers, we see these things in much higher frequencies in males. And if you think about it, it makes sense, because males only need one recessive allele on the X chromosome, because males only have one X chromosome for the condition to show. Females need two of them, two recessive alleles to show, and really we know if dad is going to have the condition or not. Because in order for the recessive allele to be passed down from dad, especially to females, you're going to know it, because dad has to be showing it. So let's look at this cross here. You got a father without hemophilia, so X big HY, and a mother that's a carrier. This is the exact same cross we just did, and we get the exact same probability with the offspring. We could substitute, if we're talking about color blindness, you could substitute in, you would C's, but C's are a little confusing because big C and little c look the same. So let's use B's for blindness. We're just using a different letter for a different condition, but it's the exact same cross that we've been doing. Okay? And you should be able to do different combinations, and that's really what the practice problems are about. Okay. Point 9 says state that a human female can be homozygous or heterozygous with respect to sex-linked genes. Well, we know that, right? So females could be homozygous. Oops. Or heterozygous. Sorry guys. Struggling here. So, or heterozygous. And that's because girls or women have two X chromosomes. Males, because we are XY, can't be homozygous or heterozygous, because remember, we're talking about sex-linked genes, we're only talking about the X chromosome. So if it's on the X chromosome, it's not on the Y chromosome, okay? Well, for these conditions anyways. Alright, and we've talked about this as well, that human females are heterozygous, carriers are heterozygous for X-linked recessive alleles, because in order to have the condition for the recessive conditions, these X-linked recessive conditions, they need to have 2 of these little h's because the recessive allele will only show in the absence of the dominant allele. So it needs to have 2, but if it only has 1, the dominant one's going to show, so the condition is not going to be shown or expressed, but there's a potential for this to be passed down to future generations. I think you get that. Predict the genotypic and phenotypic ratios of offspring of monohybrid crosses involving any of the above patterns of inheritance. These are the practice problems. So you've done one set, there's a second set with the substitute, and I would practice those and make sure you got them down. Just a couple of words to remind you. Genotype is the symbol that we use to describe the allele combination for each individual. For example, if we're talking about height in pea plants, not humans, in pea plants, a tall plant, if it's tall is dominant, could have this genomic, this genotype or this, right? And so if you count up the total number of genotypes that are possible between a combination and write them as ratios, so you can do the genotypic, and then the phenotypic, you would actually combine these two because they would both be expressed as tall plants, and count up how many tall versus short plants you have. And that's how you figure out the genotypic and phenotypic ratios of offspring. Monohybrid, mono means 1. And really, when we talk about monohybrid crosses, we're talking about this Punnett square that we only have 4 squares for because we're following one gene. Okay? So we're only following one gene. It's a monohybrid cross. The hybrid is when we cross 2 individuals or 2 sex cells. We get a hybrid. So you need to be able to do this for simple dominance, which we talked about in class last time, codominance, sex linkage, and then also with codominance, make sure you understand multiple alleles, focusing on our example of blood types. Okay, and then point 12 is deduce the genotypes and phenotypes of individuals in pedigree charts. And so here's a key for you that will help you understand how to read pedigrees, but all individuals have pedigrees because all individuals come from other individuals. So this is just basically kind of like a family tree, but it's inverted. It goes the other, well, it's a family tree, but that follows certain genetic conditions. So in this first generation, which is usually called the P generation or the parental generation, the second generation is called the F1 generation or the first filial, and then the next generation is the F2, which is the second filial. But of course, these individuals in the parental generation have parents, so it's all relative. So if we're considering this to be the parental generation, their offspring are called F1, and their offspring are called F2. But if we're looking at these individuals' parents in the parental generation, and consider them in this pedigree, then these would be the F1 and their parents would be the parental, and everything would be shifted. But we're following color blindness here. So here's an example. We know that here, and this key is going to help us, that the half colored in circle is a carrier female. And if this is for color blindness, then we know that this individual had to be X big B, X little b. And this square represents a normal male. So that would mean X big B, Y. So if they have 2 sons here, so we know that they have to have gotten their Y from dad and their X's from mom, but we know that they're normal because they're not colored in. So they must have inherited the dominant allele from mom. Now, that didn't have to be the case. They could have inherited the recessive allele just as likely, but that's not what happened in this family. But they also have a daughter here. And we don't know if it's colored in or what. That's why they left a question mark, and that's what we're trying to solve. So we do know that she must have received X big B from dad. If she's a girl, she could not have received the Y from dad. So we know that she's not a colorblind female, but we don't know if she's a carrier or a normal female. So in order to figure this out, we can go one generation further and look at her offspring. Now, her husband, or the guy that she got busy with here to have these offspring, is normal. So we know that he must be X big B Y. That means that his sons here that are unaffected must be X big B Y. The Y came from their dad, the X big B came from mom. Now this is a normal female. So she has an XX sex chromosome genotype, and she's normal, so she must have a big B, and she's not a carrier, so she must be homozygous dominant for this trait. But that still doesn't help us figure out what the other X is here. We don't know if it's a little b or a big B, but we need to look at this individual. Now this individual is a colorblind male, so he received his Y from his dad, and that means he must have received X little b from his mom. So that means that mom is a carrier just like her mom. And so she should be, oh sorry, I did the wrong half there. She should have her circle filled in halfway. Okay? So this is a carrier, and this is the genotype that we were solving for. So there's some practice problems with these as well with your substitute, and you can practice those as well, because remember that you need to be able to deduce those genotypes and phenotypes. Freshen up on what the word deduce means, and what IB expects you to be able to do with that word, okay? And that wraps up actually no, let me do one more thing. Sorry, I know that this has gone on for a long time, but you're doing this in class, so it's okay. I'm not asking you to do it outside of class. Remember that in meiosis, let's look at we'll look at metaphase 1. Let's look at metaphase 1. And we know this, if you remember, that in metaphase 1, we have chromosomes line up on the metaphase plate as tetrads and homologous pairs, because in anaphase 1, these homologous chromosomes are going to separate and get pulled apart. Remember, they're sister chromatids. Now, if we were to do some gene mapping, we might know that on this chromosome, if we're following gene A, and the sister chromatid would also have gene A, this sister chromatid, let's say the red came from mom and the blue came from dad, let's say mom contributed a recessive allele. Well, this cell is going to continue to divide so that we would end up with a cell that would say that we're developing into sperm, might have the chromosome for big A. It may have the chromosome. It may have the other sister chromatid, or it may have the little a, or its sister chromatid of a little a. And so when we do our Punnett square, what we're doing is following that gene, but because it's doubled up, we're not concerned with the double because it's equal odds. So we're looking at big A and little a, and so if the parent for this trait is heterozygous, big A and little a, we put that individual on the Punnett square, we put big A and little a on the side, because this individual, this dad, is only going to be able to contribute one of these two unless something funky like non-disjunction happens to the offspring. And the same situation is going to happen with the egg, but remember that in oogenesis, only one of the 4 cells is going to be the egg, but it's still, as far as we know, random about which one. So there's a potential for that genetic, those genetic differences. And so you really need to make sure you understand how this connects to meiosis. These Punnett squares impact meiosis, but notice and remember that there are other traits, there are other genes, other traits that are coded for by these genes. So if we're using big T and little t, that if these chromosomes travel with each other, sorry, this should be big T, this is a sister chromatid, we'll put little t's over here. This would have the little big A and big T, big A, big T, little a, little t, little a, little t. And so those genes are actually linked because they're on the same chromosome. And they'd only be separated if crossing over happens somewhere between them. And this is quite interesting because we see this happen quite a bit. We don't have an individual chromosome for every gene, but rather chromosomes are the vehicles by which the genes travel. They're kind of like the cars in which all the genes pile in, and then crossing over is kind of like, let's mix up, we need to switch some of the people from one car to another, and then their end location may differ. Okay? So I hope that makes a little bit of sense. If not, jot down some questions, we'll talk about them when I come back, okay? Make sure you're ready for the weekly quiz. The weekly quiz is on material up through 4.3.4. So this material on sex linkage is not covered in it, but you need to be able to do things like the problem set that you received from last class. So again, make sure that you looked at that answer key and you figured out what's going on there, even if you did not get it right the first time. There's probably more value in spending some time to try to figure that out. Okay? All right. See you guys next class.